Okay, hi. I'm Wendy Demas. I'm a curator for the RGD, and today I'm going to talk to you about even more data that we're starting to bring into the database. So um, we're we're bringing in more rat expression data. So RNA-seq expression values for 5 species from EBI's Expression Atlas and NCBI's, well, GEO, have been imported and they're displayed on the RGD gene pages. So if you'd like to access any expression data, what you can do is pick your favorite gene, navigate to the gene page, and then you can either, okay, um, click on the menu on the left-hand side, the expression, and it'll bring you down to that section, or you can go ahead and just scroll down and it'll bring you to the expression section. This is it. Let's see, I'm using my notes too just a little bit. I was trying to. So the data are presented on the RGD gene pages in a summary table. The expression values in the table are stratified based on the TPM value thresholds. It's the same scale utilized by the Expression Atlas, where high expression is a TPM value of greater than 1,000. Medium expression is a TPM between 11 and 1,000. Low is between 0.5 and 10, and then below cutoff is a TPM value of less than 0.5. The number of samples where expression was seen in each of these 4 categories is shown in each row, and they're grouped by system. Okay. Sorry. Users can view a detailed expression report for all organ systems by clicking on the View RNA-seq Expression Data tab. So that'll give you the data for everything, or if you're just interested in a specific organ system, you can click on one of these numbers and you'll get the expression data report for that gene. And this report is downloadable and includes information such as strain, age, sex, tissue value. It also provides a link to the reference where the data came from, what reference assembly the data were aligned to, and what unit was used, the value is. To date, a total of 31 studies encompassing about 22,000 records with an average of about 32,000 individual expression values per record are viewable in the database. Initial loads were imported from Expression Atlas, and they leveraged the standardization of data, the metadata fields, and the mapping of the sequence to the RN6 assembly. So there were 22 studies for human, 3 for rat, and 1 each for pig and vervet, imported from Expression Atlas. And then there were 3 rat studies imported from GEO, and there was another study imported from GEO that was both dog and rat data. In all cases, samples were untreated controls and expression values were in TPM to allow for comparisons across samples and across studies. The goal of the expression that I'm talking about today, the expansion of the data, is to import more comprehensive data sets that aren't limited to just untreated controls, and to also integrate those publicly available data into the larger ecosystem of the RG database so users can seamlessly query for more coherent gene information. So the expansion project is basically broken down into 3 phases. The first is, the aim of the first phase is curation of the metadata from the free text descriptions found in the GEO studies, and then we're going to also import the submitted data values from GEO. In phase 2, we are going to remap to the most current and correct rat genome assembly. And then in phase 3, we are going to display those remap data and increase the types of visualizations that we provide to users to look at expression data. So a curation software tool was developed to aid in the curation and help us standardize the metadata. And currently we have just over 1,000 studies that were imported into this tool. And so you got to start somewhere. I've assessed about 800 of the studies. And this first pass assessment just highlights the importance of this project and why we need to standardize some of these data as we pull them in. So the first thing that's clear is that when data is submitted to the data that have been submitted to GEO, there's a number of reference assemblies that are cited as what the data were aligned to. In some cases, we have the study said that it was a rat study, but when you look at the methods, it actually was aligned to mouse, or rat cells were used, and a custom reference was made. So it's a little messy trying to figure out what assembly the data were aligned to. The other thing you'll notice here in the left is that the types of data that are submitted to GEO are not standard. You have differential expression, FPKM, TPM. Sometimes you have super series which come in and they have multiple data types that are often provided. So displaying this data is a challenge because they're all in different units. The other thing to note is that not everything that's in GEO has been published. There are quite a few listed, but 20% of the 800 studies I reviewed when I was writing this talk were not published. So getting some of that extra information is a bit of a challenge. So the curation of the metadata for phase 2 begins at the project level. So this is the expression curation tool that was developed. It relies on natural language processing to match ontology terms to GeoAccession attributes. This comprehensive interface provides 7 fields for controlled vocabularies, such as Uberon, vertebrate trait, clinical measurements, experimental conditions, and more. There's also fields for age, life stage, sex, culture duration, and experimental conditions and attributes. And then also when you're in GEO, if there is a citation listed, sometimes there's multiple citations listed and associated with that GeoAccession ID. So we also built in a field to include those as part of our curation so that that data can also be linked to those studies. Okay, so where the prediction software finds a match, the original text from the GEO record, it suggests a term for the curator, and sometimes it's correct, and other times curators will need to provide terms that we're missing, or we can provide more granular terms, and our developers built in these like ontology like lookups so that we can go in and we can select the terms that are in the vocabularies. The data is pre-populated, it's verified by the curators. We also go into the descriptions from the GeoAccession page, and then we can also look at the publications. Terms are entered a single time, and then they're propagated across to all the samples where it's applicable. Okay, I practiced much faster than this. I was done in like 5 minutes. So after the project level curation is completed, then the curators are brought to the sample level curation. So on this page of the interface, curators are able to assign specific details to each sample. So in this case, skin was the suggested term. So the curator selected dermis, but there's actual epidermis and dermis samples. So that's switched on a per sample basis. And then we can put in the experimental conditions yet, but our tool doesn't quite yet assign those. So over here on the right, I don't know how well you read that, but I was able to select for like a control and experimental condition. So once all of this is correct and as much information is provided as possible, the curator will load the samples. And then at that point, the metadata are loaded into our database, and then the originally publicly submitted expression values will also be loaded for all the genes in the corresponding files. So in summary, of the 800 geoaccessions that were assessed at the time I was preparing this talk, 101 of them have been loaded via the new curation tool. Right now, we're just focusing on curating studies that have TPM and FPKM data. So 529 were categorized for future curation, 24 were categorized not for curation, usually because of the lack of species information. We've notified geo publications that are associated with IDs, and then we also did need to reach out to authors for data clarification, so we've done some outreach. The aim of phase 2 is to remap to a common and most recent assembly. So we're working on a pipeline right now and assessing that. There's some common quality control tools that are built into this workflow that we're testing. The first and I think most important test that we do is after we pull down the data from the SRA and transform it into FASTQ files, we actually verify that it's Rattus norvegicus, not Rattus rattus, not human, not something else, because that can be declared in the geo submission. Then we also, just get a basic assessment of the sequencing quality, and then we're using a 2-pass alignment with STAR. So we're also looking at the alignment quality, and then I'm using RSEM to generate TPM values. So in this study was submitted by Lee et al., and there wasn't really a lot of information on it except for that they used RM7, but I just wanted to see how my processing with this workflow compared to something that was submitted, and just to see how close I was if the pipeline's okay for starting. So basically the study started with a different number of genes, probably because they had different GTF files. And then what I did is I normalized the data by filtering to the common genes that are for the GTF file that I'm using in our workflow. And then I just wanted to compare genes across the study that were expressed. And so I looked at the FPKM data and count data that was submitted, and I used an R script to convert it to TPMs. And so far, our pipeline had a 92% overlap with the public data, but I'm still looking into it to see where those differences were, if it's by biotype, or if there's any like commonalities between the biotypes. And then the last part for phase 3 is data and visualization. And Jennifer gave a really nice talk about JBrowse 2 as part of phase 3. We do have some of the publicly available expression data already loaded in, and you can see those tracks here. But we are looking to maybe increase our visualizations and use something like displayed here by the Alliance. I'd also like to thank our team and our funding and the organizers of this meeting for letting us talk about the even more data that we're going to be putting in. And then I'm going to also make a shameless plug. Our RGD 2024 desktop calendar is going to the printer soon. If anyone would like to show off their model organisms, you have a special meeting deadline extension. Let me know if you have a picture, or you could submit your images to this email address. So thank you again for your time and attention.